So now we're going to begin our discussion of membranes. All cells have a plasma membrane that separates the cytoplasm from its surroundings. And while this provides a barrier to toxic molecules, that membrane has many other functions as well. Transport, for example, energy transduction, cell motility, reproduction, signal transduction, and communication with other cells in its vicinity. Lipid bilayers form spontaneously without the input of free energy due to the influence of the hydrophobic effect. We have seen that before in driving protein folding. So this favors the aggregation of those nonpolar groups, and once formed, these structures are quite stable. So these forces drive amphiphilic lipids or amphipathic lipids to form membranes. This also allows or forces and facilitates the association of proteins with those membranes. And we're going to look at examples of these. So biological membranes contain a varied amount of protein. It depends upon their function, essentially. Some membranes may contain as much as 75 to 80% protein, or as little as 15 to 20%. So as I said, depending upon their function, the amount of protein is going to vary. So membranes that carry out enzyme-related reactions and transport activities are going to be richer in those proteins. An example of that is the intermitochondrial membrane, and we'll look at some protein structures within that membrane. Membranes that carry out fewer such functions are richer in lipid. So the myelin sheath, which is a more protective layer. So cells are going to adjust the lipid composition of their membranes to suit what their functional needs are. So the lipid bilayer leaflets in these bilayers seldom have identical combinations. So we're going to vary protein, we're going to vary lipid content. However, on the cell's exterior, or facing the cell's exterior, glycoproteins and glycolipids with carbohydrate moieties are typically found there. These are usually involved in cell recognition. We can find sphingolipids with carbohydrate head groups in the outer leaflet. So when we look at that membrane, there's going to be an outer leaflet, inner leaflet. Phosphatidylcholine, we just discussed in our lipid discussion, is found in the plasma membrane, and it is neutral, so there's no charge on that. Phosphatidylethylamine is also neutral. That is found on the inner leaflet, PC in the outer leaflet. Phosphatidylserine is also found in that inner leaflet. Phosphatidylserine is the only component in the leaflet that we're going to look at that has a negative charge. So when we look at those PC, PE, PS, phosphatidylcholine has a negative charge, which makes that inner leaflet slightly more negative than the outer leaflet. And phosphatidylserine is found only in that inner leaflet. So what are the type of structures that are formed by these lipids in membranes? So very few lipids actually exist as monomers. In aqueous solutions, they're going to associate with other lipids to spontaneously form structures such as monolayers, bilayers, and micelles. So that's what we're going to look at right now. Monolayer in A is depicted here. Here we have an aqueous solution exposed to atmospheric air, and as you would predict, the polar, so this is the polar end of that lipid with its nonpolar tail, that polar head group is going to interact with the water and form this linear layer or monolayer. Let's go to the bilayer second. Bilayers, and here we see the aqueous on each side, so we're going to call this the inner layer and the outer layer or leaflet, but the head groups, again, polar head groups are going to associate with the aqueous side of the membrane, and then we're going to have those hydrophobic tails interacting with each other within that membrane structure. So two monolayers with hydrophobic tails interacting form that bilayer. Now micelles are a different type of structure, and it depends upon what type of solution they are in with respect to how they form. These are small structures. They're only composed of a few hundred molecules. So if we look at a micelle in water, so here we have aqueous solution, again, those lipids are going to orient themselves with that polar head group facing the water side, and we're going to have those hydrophobic tails in the middle. Now, if we have a nonpolar solution, which is what we're looking at here, and we can capture some aqueous solution here, we're going to have a different orientation. So, in nonpolar solvents, that orientation of the lipid is going to reverse. There is something called a critical micelle concentration, or CMC, and this is the minimum amount of lipid that's required to form these micelles. And here are two other structures, two other vesicles, so to speak. On the left, or in D, is a multi-layer or multilamellar vesicle, so it has several lipid structures surrounding it. Here is a unimolecular vesicle, so we just have one bilayer, so to speak, that has become a micelle. So we've got the head groups out here, and we have in the inner side we have head groups here. This is a representation, or I can give you an example of this, liposomes. I don't know if you've encountered that term in biology, but these are highly stable structures. They're closed, self-sealing, solvent-filled vesicles, and they can be used for drug delivery or delivery of DNA into cells. This is an electron micrograph of a multilamellar structure in the Golgi. So this is a common structure in some of our cellular organelles. So I know everyone's heard of the fluid mosaic model. Okay. This is a phospholipid bilayer, and it's a fluid matrix, meaning it's not stationary or not, there's flow, there's movement within that bilayer. So there can be lateral diffusion, and there can also be some flipping depending upon what this bilayer is. Components in these bilayers with respect to proteins, we can have integral or intrinsic proteins or peripheral extrinsic proteins. And we're going to take a look at some proteins that also are anchored to that bilayer through lipids. So when we talk about integral proteins, sometimes they're called icebergs floating in a two-dimensional lipid sea in a random or mosaic distribution. The integral proteins can diffuse laterally in that lipid matrix unless their movements are restricted by other cell components. Let's see. Portions of the protein in contact with the nonpolar core of that lipid bilayer are going to be dominated by alpha helices or beta sheets, because these secondary structures neutralize that highly polar peptide backbone. Peripheral proteins can bind to the membrane in several ways, and we'll look at some of those attachments, but they are loosely attached to the membrane. So if we have an integral protein, that is going to transverse the membrane, so it would require us to disrupt that membrane in order to remove that protein from it. So we really need harsh treatment to do that. Peripheral proteins can be more easily dissociated from that membrane. So most eukaryotic plasma membranes are about 50% protein, and we're going to take a look at some of the modes of attachment here for those peripheral proteins. They could be through ionic and hydrogen bonding interactions, so that's what's represented here. We get to have an amphipathic alpha helix, so we'd still have some type of interaction between, the amino acids in that protein with those polar heads. We could have hydrophobic loops. So this unstructured portion of this protein would contain hydrophobic residues. And we can also have association through an integral protein. So these are all peripheral proteins. Here we see that integral protein that transfers across that membrane. Integral proteins can have single transmembrane segments or multiple transmembrane segments. And we're going to take a look at some examples of those. So here's a protein called glycophorin A. This is found in human erythrocytes or on human erythrocytes. It is the oligosaccharides. Well, let me point out here. So here's the inside of the cell, here's the outside of the cell, and as you can see, the majority of this protein is on the exterior. And what we have here are these carbohydrate groups attached to this protein. And it is those oligosaccharide groups that are responsible for the ABO blood types. Here is our transmembrane segment. It's composed of 19 hydrophobic amino acids. If you can look at these sequences, we have glycine, isoleucine, valine, methionine, so forth. So these are nonpolar amino acids. About 10 to 30% of all transmembrane proteins are going to have single alpha helice transmembrane segments, So this particular protein is going to obviously can function, well, not this one, so to speak, but they can function as signaling molecules or immune recognition to host cells. This one in particular, again, designates your ABO blood type. This is another transmembrane protein, Bacteriorhodopsin. It has 7 transmembrane segments that are connected by short loops. So this is a light-driven proton pump that powers ATP synthesis. Each of these segments is about 20 amino acids long. This one has 7, transmembrane segments. So here are the possible ways that we can link proteins to that, or link lipids to the proteins in the fat, link lipids to proteins. So 2 types of fatty acids are found that link to proteins, myristic acid and palmitic acid. So we've just ended our lipid discussion. Myristic acid connects through an amide linkage into that into that with that protein. So this is called myristylation. Palmitic acid is linked through a thio group, which of course comes from cysteine residues. And let's see, was there anything else here? Myristylated proteins are often found in the cytosol, endoplasmic reticulum, plasma membranes, and in the nucleus. And this last slide is just a graph that indicates the number of transmembrane segments that are most common. So you see that 2 to 12, we've got a lot of proteins that use this number of transmembrane segments. Transport proteins usually have between 6 and 12, and you'll see that there are very few that go beyond this. So that concludes our lipid chapter, and we're going to move on to nucleic acids.